First of all, I would like to share best wishes of the executive committee with you. Uh, Having mentioned in the last session, we are all definitely aware of the importance of this course. You know why? Uh, I think the title of this course is quite important because it's our future. In the past, our health reports included just basic information such as grading and differentiation. But in the present, we are providing advanced prognostic information for our patients such as HER2 status, outfusion. But in the future the era of personalized medicine, we will be asked to report more, which chemo, for instance, is to be started, or if there's a chemo resistance, if there's a minimal disease, something like that. So we should get ready as a pathologist, and we should perform continuous medical education in molecular pathology, and we should be certified in this field. And finally, we should provide good medicine, of course. I have a wish, I have a dream to continue on this kind of courses here. This must be a traditional course. And finally, I thank all the organizers and all of you for participation. Thank you.
Hello, everyone. We actually listened to two phenomenal speakers. Dr. Dietel focused on the importance of molecular pathology today and what it will be bringing us tomorrow. And Dr. Jennifer Hunt also mentioned why, or she convinced us that we should be able to open up a molecular lab, provided that we have the technical training and know that it's a costly and time-consuming process. So today I'll be talking about molecular pathology, lab accreditation and regulation, as well as quality. And I would like to take this opportunity first to thank all of the organizing committee members, and also say hi and greet you all with warm welcoming, especially to those who have come from abroad, as well as those who have come from different parts of Turkey. So, given that, this is the outline of my talk. I'll be mentioning a little bit about the laboratory regulation and accreditation, and then move on to quality. We all know there is control of quality, assurance of quality, and management of it. And then there is this proficiency testing that we should participate in, and also available surveys or QC materials that we could use to achieve excellent quality, as well as look at the performance review or competence of our technologists that we have in lab. So as we all know, the physician orders, and there are different domains that an error could happen. So starting from the, as Dr. Dietel said, getting the specimen from the patient, the requisition, collection, identification, transportation, preparation, and up to this point, we called it the pre-analytical errors, and more than 90% of the errors happen to be contributed by the pre-analytical, and now we have the analysis and the post-analytical errors. And any break in this chain would actually result in patient management delay or some changes. And we all know that molecular diagnostics is actually very timely and costly, and it's really important not only to provide the right diagnosis at the right time, also this will result in the correct therapy and also preventive measures. So, as we all know, the paradigm of garbage in, garbage out, and even if we have the perfect stick, so the perfect model, if you have the improperly identified patient, then the result will be wrong. Or even if you have the best data or the properly identified patient, but if you collect it into a wrong tube, or if you order a wrong test, or if you have heparin that could perhaps inhibit the PCR process, again, you would either get no results or the results would be uninterpretable and you would delay the process. So how does all this affect you? Approximately, it is estimated that 70% of medical decisions are actually based on the lab results that we provide to the physicians. And this happens to account about 3.5 to 5% of total healthcare costs. And unfortunately, there are billions of dollars that are spent on a yearly basis on preventable adverse events. So that is the reason why it's really important to get it right the first time and every time. So what does quality mean? So it's a range of activities that will enable a molecular lab to achieve and also to maintain high levels of both accuracy and proficiency. So who's responsible for this quality? Is this that phlebotomist that we talked about, or is it the bench techs or coordinators, medical and technical directors, supervisors? In fact, it's all of us. We're all accountable for the best quality that we could offer for the safety, patient safety, as well as best patient management. So quality has different components. The quality control actually provides a feedback at the operator level that the process is in control, whereas the quality assurance reviews the performance of the process over time to be able to identify any trends and shifts or anything that is out of control, in other words. And then you have the quality management that would tie together both the quality control as well as the assurance to ensure high quality throughout any organization. So how do we achieve and maintain this high level of accuracy and proficiency? So you have the quality control that establish all the standard operating procedures for each step of the testing process that could range from specimen handling to instruments performance to validation of a new test, and etc. And then here comes the quality assessment that will try to check the efficiency of the quality control as well as define the administrative requirements, mandatory record keeping, evaluates the data, and then looks at the audits and also monitors the adherence to the SOPs. And if there's any problem, then they would come back and try to provide the corrective actions, document these very precisely, and then find the people who would be responsible to carry out any corrective actions. So for accurate, reliable, and reproducible test results, we certainly need the quality, and we need to develop and implement some methods. And of course, quality without a regulatory oversight is impossible, and we need those programs so that we could obtain a license to be able to operate in a clinical laboratory. So what are these important agencies? Well, Centers for Medicare and Medicaid Services, or CMS, in conjunction with CDC, have originally decided to develop and enforce CLIA regulations, and they have been continuing to oversee most of the regulatory activities at this point, including laboratory registration, on-site inspections, accreditation, and training. CLIA, which is under CMS, actually has been founded in 1967 for the first time, and it has been the first attempt by the federal government to be able to regulate the clinical laboratories. And that this oversight by the federal government has been expanded in 1988 to all of the clinical laboratories throughout the U.S. So not all laboratories, however, are regulated. There are certain agencies that are being exempt from that, but most of them are actually CLIA regulated. So what is the objective of the CLIA 88? So it actually has to ensure quality laboratory testing. It gives us all the qualifications that are needed for the laboratory personnel. They tell you that you have to have standard operating procedures, you have to perform verification, validation of any test results that you're going to be implementing, tests that you're going to be implementing. You should have clinical validity, as Dr. Hunt mentioned. Proficiency testing comes into play that we need to participate in, and quality control, quality assessment, and also you have to know what you should be doing with the specimen that's left behind. You have to record, and also calibrations of the instruments and etc. to be able to achieve the best quality. And this will be actually overseen by both professional organizations as well as FDA, and also some of the states, including New York, which is exceptionally stringent, which has exceptionally stringent conditions. And for California, for instance, you have to have, in addition to stringent conditions, another certificate. So the Joint Committee and CAP, AABB, PACT, and COLA are actually accreditation organizations. They set the requirements for quality in the healthcare industry. And I actually really like this, the vision statement. It says, all people should always experience the safest, the highest quality, best value healthcare across all settings. And CAP and AABB not only provides accreditation, they also provide education. They have many resources you could tap into, and CAP being the advocate for the clinical laboratories, and AABB for the blood and cellular therapy. So CAP, or College of American Pathology, it is not a regulatory body. It's approved by CMS. It's an accreditation program which is non-profit. Its requirements are actually either equivalent or even more stringent than that of the CMS's. And they provide the checklist that you would need, or general checklist, or molecular pathology lab checklist in their site. And they suggest that external inspections by them should be performed every other year. And in those years that CAP inspections are not performed, you have to have internal inspections, like cytogenetics lab inspecting molecular genetics lab, vice versa. So for quality assessment, there are, in addition to inspections, there are different components that needs to be looked into: documentation, monitorization, participation of proficiency testing, review of the performance of the people, and checking their competency levels, having a system approach, and also providing statistics. So lab inspections are actually done by CAP by highly qualified inspectors who use the CAP checklist. And inspectors are actually provided in advance with the proficiency testing performance history of that lab that they're going to go and inspect. And they have a 3-month period of unannounced basis. You know when they're going to come within those 3 months, but you don't know when exactly they'll be showing up. They could just give you a call and they would be there. So they would be looking at everything, but if you have persistent issues, then they might come back, but hopefully that would not be the case. So they would be looking at your laboratory itself, looking at the testing practice, documentation process, how your standard operating procedures are, if they are current, and they could sometimes follow a technician from A to Z while they're performing a test and either watch or ask questions. And then at the end, in addition to finalizing their report, they come up with their recommendations. And also they would provide with some deficiencies if there are any. But it's very important to really take into account their recommendations. So there are two types of lab deficiencies. Phase 1 is not as severe as phase 2. And phase 1, you just need to provide a statement that regarding the corrective action that you have taken if you had a deficiency. But in the case of phase 2, in addition to that statement, you also have to provide the appropriate document that shows not only the policy changes and procedural changes that you have implemented, but also all the supporting documents, including worksheets, patient logs, or meeting minutes, and etc. So it's a lot more thorough. So it's important, therefore, to move from this cross to check. The sooner is the better. So you could use professional guidelines that are available to you on AMP, ACMG, CAP, and CLSI websites, and they have all the checklists and also guidelines that you could make use of. And there are qualifications of the personnel that you could listen to. Dr. Hunt has actually mentioned them. So in addition to the qualifications of a director who is a pathologist, board certified, perhaps in another specialty, or a doctoral scientist with molecular pathology expertise, you have also the technical operations personnel. As she said, this person is the most valuable person almost, and so it's everything minus the medical background. So personnel need also to be educated, and as mentioned by Dr. Hunt, it's not really an easy process, but there is continuing medical laboratory education that is available that could be tapped into to achieve high quality employee performance. So what else does a molecular lab need? So molecular document control system should be in place. You should have to assess the competency of everyone that works in a specific assigned duty. And safety is also important. You should be conducting fire drills perhaps at least once a year. And very importantly, and this happens to be one of the most common deficiencies, you must have a very current CAP activity menu. So if you remove a test from your test menu, or if you add a test to your menu, those should be reflecting, those changes should be reflected in your menu. So in addition, looking at the turnaround times and monitoring those and trying to see if there's anything that you could improve is important. You should be able to demonstrate the appropriateness of the test, and also see if that test that you have is being used for the intended clinical purpose. And you should run all the statistics to see if there's any shifts that you might perhaps improve. And you should be doing the comparative analysis to see the percentages of normal and compare those to your abnormal findings, and if allele frequencies are similar to what you would expect to see in the population, and etc. So here's a document that I was provided, and we all know that refrigerator and freezer temperatures should be checked and recorded on a daily basis. And I was told during my training that you should never be using red pen and no highlighters, as those actually tend to fade over time. And you should not have any spaces. If this is a weekend, you should say that it is a weekend. And if a mistake was made, you should never cross it over several times. You should just put a single line and initial and date. Well, this is the result. So they had not circled whether this was a refrigerator or a freezer, and so many other problems that I had mentioned was already noted. In addition, I want you to, since this is a February, I mean, this needs to be recorded on a daily basis. It looks as if, you know, someone has forgotten and has recorded sometime in March, because there's no 31, date 31 in February. So, monitoring errors is quite important to be able to provide the corrective action. Here you see that the plotted specimens and the improperly filled specimens are high in number, so you would be able to provide the adequate training to those people to minimize such results. So coming to validation and verification, for high and moderate complexity testing, it is requested that you have to figure out your performance characteristics before you implement a new test or a procedure. And for FDA approved and FDA cleared tests, you do the verification. There are slight differences between verification and validation. And verification is not as thorough since all the FDA approved tests have already gone through the pre-approval process, pre-approval marketing process, as well as risk assessment. However, if you have a lab-developed test, or if there is a method that you would like to bring that has not been previously cleared by FDA, or you have modified slightly, even slightly, the FDA-approved method, then it becomes off-label, and then you have to really thoroughly validate it. So for validation purposes, you have to go through precision, accuracy, analytic sensitivity and specificity. You have to look at reference ranges, reportable ranges. So for lab-developed tests, this is really a very thorough process. And for sequencing, for instance, the reference range is the reference sequence. So you have to be inserting that information. And the reportable range would be the number of exons that you are testing. So for FDA-approved or cleared assays, it's precision, accuracy, reference range, and reportable range. And we also heard of the ACCE by Dr. Hunt. So we need to assess the accuracy as well as the reproducibility. So you have your sample that you know the genotype of, or you could use some controls. As mentioned before, how many do you need? It's really dependent on how comfortable you will be feeling. Maybe 30, maybe 40, maybe 50. So in addition to specificity and sensitivity analytically, you have to also assess, is my sample type correct? Am I going to be able to get the same result if I use the peripheral blood versus saliva? Or what is my sensitivity level, limit of detection? Or if I have heparin, how would my test would be effective? And you have to produce or reproduce within run. So you use a genotype and you run it 3 times in triplicates, or you have to do between run runs. So you take the same genotype. You some people do it for 3 times, some people 5 times. You run it on different runs, on different days, and you ask different technologists to do that. And you also, if that test is being performed by using different instruments, you should try to reproduce the results using different instruments. So validation, you have to really document everything in a very organized manner. And CAP actually has the most extensive proficiency testing available, and worldwide you could have access to the CAP PT surveys, and they have been providing well-characterized samples and control materials for you. And at the end, they put together a summary, they review the data. If there are any problems, they address those, and then they publish them. So CLIA actually requires participation in a system that validates accuracy if you have high and moderate complexity assays, and most of the molecular tests are those, as you know. And also if you're doing the provider-performed microscopy, and this should be done at least twice a year. And in this website, you would be able to download any PT providers that has been approved by CLIA, and you could also use alternative programs. So there are some surveys. Unfortunately, these are limited in number, but they're present. You could have the molecular genetics testing, pharmacogenetics, microbiology, and molecular oncology and HLA molecular typing. And DNA sequencing survey is the newest. As mentioned, compared to years ago in 1995, there were only 4 apparently, but now there are a lot more. But still, it doesn't have limited information on heterogeneous or rare mutations or rare diseases or recent discoveries. It does not allow any phenotype-genotype correlations, and it doesn't take into account the variability of the methods used. And the field is exponentially growing, and CAP and ACMG are trying to catch up with it. So trying to assess the competency of your staff, you should directly observe the tests that they're performing. You could monitor the way they are recording or reporting the results. You could review the results obtained by their PT or any preventive maintenance records that they take. You could look at their instrument maintenance, how they perform, and also you could ask them to test previously analyzed samples and also assess their troubleshooting skills. So in addition to internal performance characteristics, you could also look at some committees and networks, external QA and performance trends. And I just want to give a few slides on how quality at Geisinger Medical Laboratories, a hospital system where I belong to at this point in Pennsylvania, that is in Pennsylvania. So here, what we do have is we know where the quality information is. We could go to GML portal, and we have all the information that is necessary there. And then we're able to communicate with our staff all the error rates, not only the error rates, of course, the success rates also. Look at the quarterly update. We have the dashboards there, and then the PT summaries, variance reports are there. So by looking at all these different parameters, you could define new projects that would allow you to improve. And one of the things that we have is the tracking system for pre-analytical, analytical, as well as post-analytical phases, and try to see how much of it impacts and at what level. So for this purpose, there is a morning report at 7:30 a.m. Many people gather together to talk about any lab production problems, software problems, identify contributing factors, and then they get together to come up with solutions and then follow up on those solutions. So the goal is, how can we do it right the first time and every time? So here's the morning report I mentioned. There are different information, like in this case, there's a phlebotomy problem, and here you could, again, train this phlebotomist, etc. And in addition to daily reports, you have the monthly reports, and you could look at your error rates to be able to fix to ensure high quality. So in summary, you have to promote accuracy and efficiency, assess the assay reproducibility, look at your staff's competency, and try to address and institute some measures, not after things happen, but before the problems occur. And also, please listen to your inspectors and document everything in detail. So following guidelines, SOPs will allow you to ensure quality, as well as having rigid quality controls, developing quality processes to be monitored, and identifying any problem areas, fixing and monitoring. So you have quality planning, quality control, assurance, and improvement. And you have the quality standards that the CLIA will have many very valuable information for you to ensure accurate, reliable, and timely patient results. and you would have the CLSI standards and guidelines in addition to CAP to get all the focus checklists. So the other issue is actually to be able to see the patient that is hidden in that specimen, and that will actually allow you to see the hidden stories, and that every specimen is not a number but a patient. So it is really a huge elephant, so quality, but hopefully you will be able to use all the measures, guidelines, SOPs, etc. And when the time comes, the examining, quality examining people will be happy. So you'll have success in your standards, general excellence in your standards, accuracy and reliability at its highest level. That would allow you to meet all the expectations of your patients, physicians, as well as the labs that you work for. And you, by this way, avoid any incorrect, unnecessary treatments or delays in the treatment, wasted time, efforts, if unnecessary or additional testing have to be done, and above all, you decrease the cost and you maintain your accreditation. So thank you very much indeed. And molecular diagnostics, once was rare, is becoming the gold standard in most cases all over the world, and it's not as easy as it is in this cartoon. Thank you.